I think I didn't record groups 5 to 8. I'm very sorry. I forgot to reactivate the camcorder.
Yeah, welcome to our presentation here today. Mark and me are going to present today from the MOM Consultancy Group the production of possibilities of PHA from wastewater streams. It's a project we're doing with Brabantse Delta, as some people know. For our presentation, it's going to be a bit different than from all the other presentations we have seen so far. We are not only going to present the results from the experiments we did in the lab, but we're also going to have a lot of other practical work we're doing during our project, which is contacting companies to actually get like wastewater samples. And we also want to tell you something about this, because so far we haven't really done a lot in the lab, as we are waiting for the samples we are getting from the companies.
Yeah, first of all, the content. We have the introduction of the project, and the process that we made, and the selection of the companies, contacting companies, and the lab work. And then we speak about our current activities and follow-up. Yes, our client is Brabantse Delta. They run a water treatment plant, and many companies discharge their water to their treatment plant, and some of them have fatty acids in their wastewater streams. And these fatty acids can be a building block for the PHA production. So that's what we want, and we need to select the companies that have this building block, contact them to collect their waste streams, and make experiments to find out if we can produce PHA from that wastewater that we collected. Yeah, to produce PHA we need specific conditions, and we did not know them before we started the project, so we need to define the optimal conditions for the experiment, and we also made already a first selection of the companies.
Yeah, as Mark already said, we made the selection on the companies, and this selection is depending on certain data we got from Brabantse Delta. Brabantse Delta has like more than 100 companies which are actually discharging on different wastewater treatment plants. In our case, we mainly concentrate on the wastewater treatment plant in Bath, which is located in Zeeland. The information we got from Brabantse Delta was information about the COD, nitrogen, and phosphate conditions. What we found out in our literature study was that we need very, very low amounts of nitrogen and phosphate, but a very high concentration of COD. And this is like in a specific ratio, which maybe Mark is going to explain later on. So what we have here, this is like a list from the top 13 companies we selected from our list, with a whole bunch of information, and this list showed the best numbers for which could be suitable for PHA production from the wastewater streams. We see here, for example, for a company which is called Stahl in Baalwijk, they have a very, very high COD concentration and a relatively low phosphate concentration. So far about this. So the contacting of the companies, this is very interesting, I think very different from all the other projects we've seen so far here. For this we need to make a communication strategy. So first you have to make a list, and you know, okay, these 13 companies, we need to contact them, but how are we going to contact them? Do we send them a letter? Do we call them immediately? So for this it was necessary to really structure and say like, okay, we're going to do this and this steps to actually contact the companies, and in the end get a sample from them to do research about the different suitabilities for the wastewater streams.
Yes, what we also did was defining the conditions. We had like 6 conditions from a literature study, but we did not know which is really working. So yeah, we made the experiment, and produce PHA, and we have to see whether there is PHA in the solution, so we colored it. And if the solution is red, then there's PHA produced, and then these are the right conditions. And that was the case for the conditions, for this condition. So we had a pH of 8, temperature of 20, and like 700 milligrams per liter of carbon source. Yeah, that's it.
Maybe important to add, this was the first research we did in the lab, or actually the only research we did so far. And it was with the collection of all the wastewater which is discharged at the wastewater treatment plant. So you just take the sludge from the wastewater treatment plant. But what we want to do is we want to only go to the companies and see which waste streams are best suitable for them. Because everybody's now discharging on them, but from the list of 100 companies, only a few are maybe suitable for the PHA production. And this is something we still need to do. So we contacted the companies, and for some companies we collected wastewater in the meanwhile. Brabantse Delta asked us to make a recording about the response, how they reacted on our research, and whether they are interested, whether they are not interested. So we did a lot of calling and going to the companies, more than we actually did in the lab so far. After we have the samples, we can do the PHA production in the lab, as it worked out, like Mark said already in the first experiment. Can you maybe explain a bit more how we do the experiments, or how we're planning to do this?
To produce PHA?
Yeah.
Yeah, we take like sludge from the water treatment plant, then we take the wastewater we got from the companies, mix it together. And
but there was also something with the fatty acids, right? Like that you can, how to calculate the quantity of PHA.
Yeah, that's something we also want to do, but that's still in the development.
Okay.
That's
Okay. As you can see, we're kind of split up our parts because it's like very, very broad. It takes a lot of time to do things, so that's why Myra and Mark are mainly doing the lab, and Luke and me are contacting the companies, go there, collect the wastewater samples. So that's what has been done so far. Then we still need to contact the remaining companies we haven't contacted so far. Some companies are very, very busy, and they're like slow in their response. There's, for example, one company. It's I don't think I should mention the name, but they take very, very long. We called them already 7 times, but they're never available. So it also sometimes looks for us that some companies might be not interested, but overall, the response of the companies is very, very positive. They really like to see that young students are working on an applied project, which is very, very nice to see. We have been, for example, at Kapafrandra, which is like a cardboard, exactly, a cardboard factory, where we have seen like how they reuse paper and produce cardboard from it. And so it's actually very nice. You go to these companies, you see how different processes are working, and actually inform them about our project, what we're doing. And this is also a project which is not only like for us, what we are doing now. There's going to be a lot of follow-up, as it obviously takes a lot of time. We're just like one of the people who kind of creates a basement for it, or like some startup situation where an intern could continue with this work. So we don't really have a lot of research in the lab. It's overall very, very, low.
Okay. Thank you. Any questions? No? One remark, if you start saying, well, we're going to produce PAH, it's good maybe to explain first what that is for the first time that you use the abbreviation in your presentation. And I was wondering, what's the idea? So they want to see which companies have effluent that is useful to produce PAH.
PHA, yes.
Because now all of these effluents are gathered together and they go into the wastewater treatment plant, I think.
Yeah.
Is the idea that the companies that are suitable, that their effluent doesn't go into the regular wastewater treatment plant, but that they are gathered somewhere else to produce PAH. I'm not
this
sure.
could be an idea that they collect it maybe first and then use it as an input for the PHA production.
Okay.
So they separate the...
They separate it from the rest of the wastewater treatment plant.
That could be one idea. We're developing this, and then there could be also other ways to do it maybe.
Okay.
So we can maybe also produce it somewhere close to the company or something.
Yeah, yeah.
Could be
Yeah.
also the way, then you don't have to transport it.
Yeah.
Well,
But then that should be a cooperation, I think, between the company and Brabantse Delta in this case.
yes.
Because when you do it near the company, it would become more or less the responsibility of the company and not of Brabantse Delta.
And
I
actually,
assume.
that's also what's happening right now. A lot
Yeah.
of companies have already their own wastewater treatment.
Yeah.
And of course, Brabantse Delta kind of wants to avoid this because they pay a lot of money to Brabantse Delta
Yeah.
to clean up their wastewater. And if the companies start to have their own wastewater systems, they actually not discharging anymore.
They lose money.
They lose money, and that's why a reason to produce PHA could be an option where they lower the costs of discharging to Brabantse Delta.
Okay. Yeah.
Yeah.
So
this was also a point where a
strange
lot of
that
companies
Brabantse
said,
Delta would object to the fact that companies produce less waste, but then of course it's still something commercial. Yeah.
Yeah.
Okay. Thank you very much. Last but not least, Group 10.
Does somebody know how I log out?
I think you have to click allow. Yeah.
Okay, so we're the Alpha Consultancy Group, and we're doing, as you might have guessed, the same project as the previous group. So we're doing it in cooperation with the Brabantse Delta to find the possibilities for PHA bioplastic production out of wastewater streams. No.
There's an arrow at the bottom. Yeah.
Oh, yeah. So in the lab, we are trying to get these conditions for the PHA production. So we have pH between 6 and a value of 8. We keep it at room temperature. The COD should be higher than 3 grams per liter. The phosphorus concentration 0.01 grams per liter. Total nitrogen 0.05 grams per liter, and a retention time of 7 days. So first we did a PHA production trial. Here we used the sludge from the wastewater plant. We used sodium acetate as carbon source. We added a growth medium, adjusted the pH, and we put it under anaerobic conditions in a water bath at 20 degrees Celsius, and we left it for 7 days, but actually we left it for 6. So we made 8 samples, and we changed the ratio between the wastewater and the sludge. In this case, we just used demi-water as was the trial, and we changed the carbon source. I will add that something went wrong. We added 5 times too much than we should. Next slide. So after 7 days, we needed to see if there was any PHA production. We did this by using the now red dye, and by using the microscope, the fluorescent microscope, we came with some pictures after this one, which looked like this. Basically, the red in the picture is PHA, right? Or the
Yeah,
dots?
just few small red dots that you can
Yeah, I
really
think
see.
it's this and
No, just
No?
very small dots. It's not big for you.
no, there was very little, but
So which one is the PAH?
It's not really.
it's not really feasible
Yeah, on this picture.
in this picture. But there were like teeny tiny dots
Okay.
in there. But yeah, so there's PHA production already in the wastewater sludge.
here.
From the
So where are these pictures from? From the original sludge or from
the
your?
sludge.
Okay.
From the
Sludge and
wastewater
the
treatment
water.
plant.
Okay.
The first experiment that we did.
Yeah.
And it's kind of difficult to see the PHA like with this microscope. So Japanese are still looking for methods. So
Yeah, they don't quite know how to work with microscope either.
Okay.
So-
Because if you have a picture like this, how would you determine the amount of PAH?
Yeah, we
You
cannot-
have it to measure the surface area,
This
or?
is just to see if there is any
Oh,
PHA.
okay.
And if there is, then we can do a test for the VFAs, and that will determine how much PHA is.
Okay.
So, yeah.
Yeah.
and we also have some now the part of the samples of some companies because the first experiment was just the water. And then we selected some companies and the other will explain
Yeah,
it.
this is from our samples.
Yes. So then we needed to characterize the wastewater by knowing the pH, the COD, the phosphorus, and nitrogen. We also adjusted pH for a neutral one to be better production, have a better production of PHA. This led to wastewater ratio was 1:2 and 7 days retention time on shaker and still there, so we're still waiting. And then you're going to do the coloring. Actually, we already did the coloring, but just to see if there's some.
Yeah, we did this last Tuesday, but there were no real results with it either.
Yeah, because I didn't have the 7 days yet. And you use it for pH with pH meter, COD is test here.
Half
Yes. And actually in the phosphorus that I recommend to the photometric analysis. I think it's what we use it. And for the COD test, we needed to do it twice because for the first time it was too concentrated and it was too dark, so we could not really know the amount of COD. And then we did it again.
Yeah.
Yes.
The results, because we had some samples, this is for the ion chromatography, for nitrogen and phosphorus. For this project, we need a very small amount of concentration of nitrogen and phosphorus to produce the PHA, so most of them we cannot even read it. And this is a micro.
I can't understand you very well. Can you speak up a little bit?
Sorry. These values here for nitrogen and phosphorus, for this project, we selected some companies that they have very low concentration of nitrogen and phosphorus because it's required for the PHA production.
Yeah, and
So-
these were not detectable by
Yeah.
the machine.
Some of
Okay.
them could not be even detectable for the text ion chromatography. And for this company, we had 3 companies and different, actually, 2 companies, but 3 different streams. This is acetic one, because they produce vinegar.
Sorry?
They produce vinegar.
Yeah, vinegar.
Yeah. And 2 wastewater streams from different companies. And the next steps, we need to check if the PHA is produced by the fluorescence microscope. We need to measure the VFAs and measure the amount of PHA produced. But we are still looking for methods to do it.
Yeah, basically Jap and Isha are discussing this. Yeah, are there any questions?
Yes. You said that the first sample that you measured was not a wastewater sample.
No, it was not. It was the sludge from the wastewater treatment plant.
Okay.
So it was just collected over there with all the wastewater streams in it.
And you said we left it for a few days and under certain conditions, and then we measured if there was PAH production.
Yeah, this is just to have a baseline description. So if there's any PHA production in the streams already, so from the sludge, and
Oh, from the sludge itself?
Yeah.
Okay, yeah.
And to see which was the best way to use the conditions, because we
Yeah.
tried two different.
Okay. You said that there was a growth medium present.
Yes.
What is that for and what is it?
This growth medium is because we add just the new water and not wastewater, so there was no nutrients there. And then this growth
Okay.
medium just
Okay, so you had to add nitrates and phosphates and
Yeah.
stuff like that. Okay. And at the end you said that you were going to measure the volatile fatty acids, but you didn't mention the four, so what's their relation to PAH production? Or is it a
it's the
PAH or...
carbon source.
pH.
I'm sorry?
It's a carbon source, right, for the bacteria to produce the pH. So that's what they store.
We thought of measuring the VFAs before, but since it's wastewater, we did not have a method to get it into the gas chromatography.
Yeah.
Because it needs to be in the gas phase. But I think Isha and Jappe found out a method now, so that now we can measure the VFAs.
So it's the volatile fatty acids in your wastewater that are going to produce the PAH.
It's the
Of the
bacteria.
PHA.
It's consumed by the bacteria and the bacteria
Oh, that's
will
the
get
their carbon
the-
source.
Yeah.
Okay, and you used in your first experiment the
Sodium.
citrate, or yeah,
Sodium
sodium citrate, yeah, as a carbon source. Okay. Can you go back to the slide with the results for the different, that one, yeah. So what did acetic mean?
Basically, on the clone they produce die potjes van hak
Yeah,
and the conservation stuff in it. So
Yeah,
they
yeah.
have a vinegar restaurant.
oh yeah,
It is quite acidic.
yeah,
So they have two different waste streams.
okay,
Which we are investigating.
okay. And so, can you explain why the phosphorus one in one of these samples is so high?
It's prevented from Melle.
No, in the Van der Kroon you have wastewater, and in the first two where you diluted 0 times
the
and 2
nitrogen,
times.
Nitrogen.
No, on the
No,
right it says under phosphorus
Here.
2,5305. And you diluted it 5 times. So you dilute it and then you find a concentration, and if you don't dilute it, you do not find a concentration.
Yeah, we still need to talk to Jaap about it because this is also weird here.
Maybe
We
it's
diluted
like the
it
concentration of
different times.
phosphorus is too high for your... Is that possible? That the concentration in the first two is too high to measure instead of too low? Because if you dilute it further, you get the result, and if you don't...
Jaap just said that NA means that it's too low to measure. So we didn't talk to him yet because we
Did
got
you?
this
Did you?
yesterday.
Was it very blue
No.
Don't you have the graphs in here anymore?
for the phosphorus? For the phosphorus, you used the Hachlange test.
We
Just from
we
the-
just prepared the samples and then Jaap did.
This was the iron
chromatography.
Oh, it was the iron chromatograph.
Yeah.
Oh, okay. Yeah, and I'm sorry, I thought it was the Hachlange.
No.
So it's very strange that if you dilute it, that you do find a number, and if you don't
Yeah.
dilute it, you don't find a number.
Yeah,
Yeah.
because we got his results yesterday or the day before, and we're still
Okay.
not sure because of this as well. And here we don't have any value for nitrogen here,
Okay. Yeah.
so we still need to ask what. and what it
Okay.
means.
Yeah. Thank you very much. So thank you all for giving your presentations. Please remember to upload them using the assignment link on Blackboard. We would like to archive all the presentations. And I hope you have a nice holiday. I hope the weather isn't too bad. And see you again after the holidays. Yes, but you couldn't use it. I understand. From Nina, she sent me an email.